Okay, so today it's just going to be just spitballing the entire time because all of this is a little hand wavy, bioinformatics, what have you. So I'm not going to do an official, like, highly structured video for this one. So what I'm going to do is I'm going to discuss how we talked in the last video about how viruses specifically influenza and the original SARS coronavirus, were able to create peptides or create small bits of proteins that were able to trick the immune system into attacking its own biochemicals or its own hormones. So I want to, in that previous paper, we didn't know exactly which parts of the SARS coronavirus was actually doing this effect. So they basically said they said there were 6 important residues, amino acid residues, and if you have at least 3 of those amino acid residues that are lined up in the correct location on a protein, then theoretically those 3 amino acids, 3 or more, would be sufficient at creating an antibody that has at least a little bit of auto or self-attacking or autoimmunity response. I want to go a little bit further off of that. I don't want to talk about that specifically. What I would want to talk about is uh, the virus being a little bit more, what would the word be, specific than just having 3 random amino acid residues. So I guess my question is are the viruses in general uh, actually copying a lot of the genes or a lot of the structures, not just randomly throughout its genome, but is there a set location within the virus protein structures that is doing this explicitly? So here I got out the protein of the ACTH molecule. So remember, ACTH or corticotropin was the peptide molecule that your body releases, one, to suppress your immune system a little bit, and then two, it is also there to end up releasing corticosteroids, so steroids that are coming from your adrenal cortex. So this is the full length protein. So you have your small ACTH peptide, but that is a part of a larger protein. And this large protein makes a lot of peptides that are used in cellular signaling, and they are used as hormones after they've been cleaved, broken apart, and separated. So here it's called pro-opiomelanocortin. So this is encapsulating much of the what would the word be? Much of the responses that the peptides that are contained within this protein do. So some of the peptides will interact with your opium receptors. So when people take opiates, they are emulating what some of these peptides are doing, and so that's one of the reasons why opiates are so addictive, is that they are uh, basically replacing some of the peptide hormones that are in your body to try to elicit a very strong response. So one of those would be endorphins. So endorphins interact with the opiate receptors in your brain, and I believe you have them throughout your entire body as well. So beta endorphin is also included in this molecule, and then there's also the melano, because some of the peptides are associated with tanning and the activation of melanocytes. So this is where melanin comes from. So if you have more melanin, right? How do I phrase that? So basically, by producing these peptides and releasing them, you are in fact encouraging the development of melanin. So, and this is a somewhat controlled process because when you are not getting enough sunlight and you're not producing enough vitamin D, you do not want to be producing melanin. That way, your skin becomes more vulnerable to the sunlight, and then you can make more vitamin D. But if you're getting plenty of sunlight, you're going to want to produce some melanin to protect your skin from the sun because it's a balancing act between the sun's rays damaging you and the sun's rays producing the precursors to vitamin D. So there's that, and then there's also the cortin, which is the part that um, produces the corticotropin. So going down, and this is at Yon Approach, which is an excellent database of a lot of proteins found throughout nature, and they have really good tools. So where you can put in the protein structures or the protein amino acid sequences, and you can align them with each other, which is what I'm going to show you later, or you can blast them, which is when it would be sort of like a Google search of the entire protein or the protein amino acid sequence, where it'll go through the entire database of what is known about the amino acid sequences and try to find parts of other other amino acid sequences that align with the protein that you have provided, or the amino acid sequence that you have provided. It doesn't always work. The algorithms to do such a thing are all they're all different, and they all have their own limitations. But especially with the alignment, there are some problems with the alignment that I don't that make it very difficult to um, to extrapolate on. But it is excellent nonetheless, even with its own limitations. So, continuing down here, they have the parts of the protein. So here is the full-length protein amino acid sequence. And here they're saying the molecule gets processed or gets proteolytically cleaved into different peptides. And so you can see these different parts of the protein are encoding for different peptides used for cellular signaling. So here you have the beta endorphin, which is the latter part. Here you have melanocyte stimulating hormone beta, which is right here. Here you have lipotropin beta, which is a longer one, which encapsulates all these other peptides. So I need to also mention that sometimes you can get a full peptide, and then within that full peptide, there are smaller peptides that are completely different hormones. So here we have the corticotropin, which this is one of the more important ones that I want you to focus on because 138 to 176, these are theoretically the peptides that are being sequestered by antibodies that your body produces in defense against the virus or the original SARS coronavirus. But then also this happens with influenza, and it also happens with the newer coronavirus as well. Don't quote me on that, but they're similar enough where any effect that happens on any antibodies that are being produced against SARS are probably going to be a little bit cross-reactive with SARS-CoV-2. So what I did was I didn't just take this protein and did an alignment on it. I took this protein and I found three pro-opiomelanocortin peptides or proteins from other species, and I lined them up, and then I aligned those with the spike coronavirus proteins of 4 different coronaviruses. So I did the SARS-CoV-2 spike, I did the original SARS spike, and then I did, I think, a bat coronavirus and maybe a feline coronavirus, if I remember correctly. And so here are the results. So I'll X that out. So here we have the results. Now let me back it up a little bit. Oh, I have so many of these open that if I change the size of them, it has to change the size in all 20. So here you can see the amino acid sequences, and here is the spike of the SARS-CoV-2. Here's the original SARS-CoV spike. And remember, the spike protein is a protein on the surface of the virus that is used for gaining entry into the cell or the susceptible cell. And then here is the, yeah, this is either the feline or the bat, and this one is either the feline or the bat. Oh wait, this is not the one I wanted. Where is it? So this one, I removed one of the cat things. So, you know what? Oh, here we go. Here it is. So here, I have the 4 spikes, and then I have the 4 pro-opiomelanocortin proproteins. And here is the human one. So remember, this line is the human line. This is the gorilla one. This is a different monkey. And then this is a cat one. So, and I included cat because I do know that cats are also able to be infected by the SARS coronaviruses, SARS and SARS-like coronaviruses. So here, they line up the proteins, and what I found interesting was that the entire protein was able to be lined up in just 2 sections. So this is one of the weaknesses. One of the weaknesses that I find with this particular search or align algorithm is that it goes in order. So a problem that you might run into is what if the protein that you're wanting to search got rearranged so that let's say the protein has A and B, and then somehow it got rearranged so that it's B and A. And if you try to align that, it will only be able to align either A or B with the original, with whatever sequence you're trying to align it with. But I should also mention that a lot of this is hand wavy. A lot of this is, what would the word be? You could probably just randomly type in letters and find a way to align them, right? So random letters, by chance, will have alignments take place. So what you really need to look for when you're doing these types of alignments is, do you see significant number of absolutely conserved residues in an area that is not, what would the word be? Where there is not a lot of shifting that has to take place. So for instance, here we have, this is probably most likely just random chance creating these particular alignments, because perhaps there wasn't anywhere in the entire genome or the entire amino acid sequence that it could align with well. So they just put them here because it was somewhat similar enough. And the same might be said for this portion over here. Granted, to me, this is a little bit more robust than a lot of other things that I've seen, because I've done other protein sequences where I try to align them, and they don't usually end up this cleanly. So a lot of times, you have a lot more of these going on, and when you have a lot of these going on, then you're basically being told by the alignment algorithm that it's basically just trying to align these amino acids any which way that it can, because there is no true alignment going on. But in this particular case, there seems to be significant and ordered alignment. So here, here, this is probably close enough to actually have some sort of physiological purpose, or at least if you're making antibodies against this, they are probably going to be able to somewhat cross-react, especially since, well, in this case, the peptide, the first peptide, is going to be this, which is the signaling peptide, and then here on down, at least for the uh, signaling protein. What was I gonna say? Uh, you know what? Just continuing on. I just want people to be aware that a lot of this stuff is very easy to. It's very easy to see patterns where patterns do not exist, and that's just human nature, right? Humans try to find patterns in places that they don't exist, but that's what antibodies do as well. Antibodies try to find patterns even when that pattern shouldn't exist, right? So an antibody just has a certain set of amino acids and shapes within it, and it's just trying to find the pattern of that amino acid, which is why autoimmunity is such a problematic problem, because it is also very hand wavy. Antibodies are also very, I'm just going to find the thing that the pattern that I want to see. And if I find that pattern any which way, then I'm going to feel fulfilled in my life. So antibodies, I guess, a microcosm, if you will, of the human condition. So continuing, I did this alignment, and what I found nice was that it aligned in the two pretty nice areas. So rather than having to go through the entire sequence, I could just limit it to just two blocks. So continuing on, what I see here, is certain areas and certain peptides where I get conserved, absolutely conserved residues for multiple sites, and then very similar residues for the other sites. And keep in mind, these, and I have the peptides highlighted in purple, these peptides are going to be secreted and released into the body. They are hormones. So if there is enough similarity between a piece of your spike protein that ends up getting, let's say it's getting degraded and it's getting released, because degradation is a part of the virus life cycle, you can theoretically imagine an antibody, if it's being produced to attack these sequences, then it could very likely attack these sequences as well, especially if you have 1, 2, 3 pretty much exact matches. So here there's a little bit of there's a one gap going on, but to me that's probably close enough, especially since there is enough distance here where one being off by one probably is not going to affect the antibody binding that greatly. So, and I also did another search of these sequences with another program that was provided by Harvard, and it shows that this is actually a relevant amino acid sequence in both, or at least in peptide studies. So there's this, and then continuing on. So here we have a lot of areas where we're going through the spike protein, but not really going through the pro opiomelanocortin proteins. But because the pro-opiomelanocortin protein is much smaller than the spike proteins. And I also want to mention that I'm using the spike proteins because they are the proteins that are going to be produced at a to a really large degree. So there might also be quite a lot of protein that is synthesized as the polymerase, the RDRP, but those proteins are not going to uh, be as abundant, and the especially um, on a per molecule scale, they're definitely not, excuse me, not going to be as abundant. But with the spike proteins, there's going to be a lot of those being produced, and a lot of them are going to be catalytically cleaved as well. So I just want to bring attention to this particular sequence with quite a bit of homology. And then I also want to bring some attention to this sequence right here, because what would be very important for this case is that this is the cleavage site of the spike protein between the uh, S1 subunit and the S2 subunit of the spike protein. So this is the cleavage site, and here we have our monobasic cleavage sites for the three other coronaviruses, and then we have the polybasic or the furin cleavage site for the spike protein. This is where the spike protein is going to be cleaved, and what we do know in the in some of the studies where they have synthesized fake, I shouldn't say fake, but where we have synthesized the weakened viruses that express the spike uh, protein from either the old coronavirus or the new coronavirus, we have seen that in a lot of the fake viruses, this entire S sub 2 subunit down here has already been cleaved when the virus is formed. So for the old coronavirus, the entire spike protein, for the most part, was whole in the virus. But for the SARS-CoV-2 spike protein, this entire part, so here, 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 all the way down, this was already separated. Now, we believe that they were still touching each other, but not covalently bound in the end virus. So what this can tell you is that all of this might very well be processed within the cell into smaller peptides. And especially near the end when the cell's dying and it can't complete the full degradation process of these peptides, a lot of them are going to leak out into the environment. Because remember, a cell that is getting infected with a virus, all of its functions are, I shouldn't say all, but a lot of its functions are being redirected towards virus production. And then the cell basically just dies, and everything within the cell starts leaking out at the very end. So I want to bring attention to this because this cleavage site of the spike protein very frequently, or I shouldn't say very frequently, when I've done this alignment multiple times, and these types of alignments correspond with one of the cleavage sites of the protein, of the pro-opiomelanocortin protein. So the protein usually uses these dibasic lysine and arginine sites to denote locations that it is going to cleave into its constituent peptides. So here I will do KRs. So here you can see in the proteins that you have these KR sites. And these KR sites are how your enzymes recognize where to do cleavages in the protein. However, not all of these sites are cleaved in the active peptides, but this is the signals that they use to tell them where to cleave. And so here, these KRs are a part of the spike protein, not the uh, opiomelanocortin protein. Or I'll call it P POM. I'll call it POMC from here on out. So this is okay. Continuing on with this particular cleavage site of the spike protein, and it corresponds with the cleavage site of the part of the POMC which denotes which encodes for the corticotropin peptide. So here, this peptide right here is the corticotropin peptide that is so frequently used or so frequently targeted by your own antibodies. So what this tells me is that this, well, if we're going to get really hand wavy, what it sounds like to me is very, very, very long ago in virus evolution, this virus somehow took the entire pro-opiomelanocortin protein and then conserved its own cleavage sites and used one of those cleavage sites to be the same cleavage site of its spike protein. Now that's extremely hand wavy. Don't take that to the bank. But I mean, it's sort of with the ordering that's going on. That's sort of how it speaks to me, and I might be seeing patterns where they don't exist. So just keep that in mind as well. But to me, that would be a very effective strategy of a virus, where if a virus wanted to create a protein that has a very specific cleavage site, then it would maintain that cleavage site or reutilize that cleavage site for its own purposes. Or the reverse could have happened way long ago in evolution as well, where you had a virus that infected a creature, and granted it might have been a very, very, very primitive creature, and then somehow this creature was like, I like this virus protein, because this virus protein, like as it breaks down into these peptides that are very useful for me. So then it can say, okay, I want to keep this virus protein, and then over time it mutates the virus protein, and then it will add or remove some of the cleavage sites, and then you end up with a completely different peptide sequence that your body uses for creating hormones. That's all hand wavy, but to me, the ordering and the conservation of at least some areas, that it speaks to me on a spiritual level, right? So 28 minutes. So what I did was I did this just to I aligned multiple sequences together, in case, just to remove any types of biases that an individual species might have. So some species might have created significant numbers of mutations to infect a different host, for instance, and I wanted to sort of erase those types of mutations to see what is being conserved, and in the conserved areas, how well do they align up. And so here I'm seeing an entire area where there is pretty good, at least chemical similarity throughout the entire thing. And then I also see, only one shift going on in this entire sequence. And then I also see a large number of conserved residues. So here, if you could imagine, you have 1, 2, 3, 4, 5 absolutely conserved residues out of 1, 2, 3, 4, 5, 6, 7, 8, 9. So 5 out of 9 that are lined up perfectly. And plus, some of these mutations are very similar. So going between phenylalanine and tyrosine, that is only, for those of you who don't know, phenylalanine is an aromatic residue, and then it's a very large plate-like structure, and that's phenylalanine, and then tyrosine has just a little alcohol group sticking at the end of it. So those are extremely, extremely similar. Um, and this is also happening at one of the cleavage sites. And if I remember correctly, um, this site is important for this. These peptides are opioid antagonists or agonists. So this amino acid sequence can cause some influence on the opioid receptors in your body. And if I remember correctly, so let's double check where the beta endorphin, so this is about 237. So 236, 237 on the sequence. And so if we go to 237, we can see, yes, so this is the starting group of beta endorphin. So here, 237 is starting here. This is beta endorphin starting right here. And if you could imagine, beta endorphin, it's sharing a lot of the same sequence. And what is especially, what I will definitely want to note is here we have tyrosine, glycine, phenylalanine, blank, blank, and then serine. So here we have, especially if this were to form an alpha helix, then an alpha helix is when the amino acid sequence is making a helix. If you have one amino acid and then two more, the third amino acid will be close to the first one. Or I should say 1, 2, 3, and then so 1 and 4 will be close together. And then by proxy, 4 or 5 will be close to each other. And then if you do another 2, then it'll be 6, 7, and 8 that are close to each other. So here we see a tyrosine, a phenylalanine, and a serine, pretty darn conserved, and then absolutely conserved. And if it's on a alpha helix, then these will end up being on the same part or the same side of a helix. So if you're making an antibody that were to attack this location, and granted, I'm assuming it forms an alpha helix, or at least part-time forms an alpha helix, then there will probably be very good cross-reactivity between those two amino acid sequences. So this is just taking all of them, and so what I'm going to do is I'm going to focus my attention on just doing the spike SARS coronavirus and the human POMC. So, you know, I think we have done enough here. So here we have the PRAR. And, oh, I should also mention, so this was the site. This is the furin cleavage site that a lot of people said, oh man, it was introduced into the SARS coronavirus because it's not in any of the close relatives of the SARS coronavirus. But it actually aligns better with the pro-opiomelanocortin protein than the other nearby coronaviruses. So, I mean, what this would tell me specifically, how this would speak to me, is perhaps the removal of it was more like maybe this was the original form, or the closer to the original form of the virus, and then over time it got mutated out so that the virus could be more specific. You know, that's just the type of what would the word be? The type of qualitative experimentation or the qualitative analyses that we can do on this. So, yeah, I did the same thing with just these two, but I don't think I can add anything more. Just saying that I did have one site with pretty significant homology and early on, which was the same for the previous protein, and then I have one site Oops, I must have accidentally clicked something that I didn't mean to. So there's this, and what I did following this was let's go with the presumption. Let's go with the presumption that the spike protein, at least, is trying to recreate a lot of the peptides that can be found in the human body, especially ones that are utilized for producing antibodies. So what I went to was PepBank, which is, excuse me. So PepBank is very similar to this Yon Approach database. But what they do is you can type in amino acid sequences and they can search in their database to see if similar amino acids have been used in research or have been used uh, yeah, basically in research, or if they've been discovered in any way, shape, or form. So what I wanted to do, and I'm gonna have to zoom out a little bit so that you can see it, was I did this with the spike protein, and then I also did it with the RDRP, or the RNA-dependent RNA polymerase. And what I found, which this is 1,273 amino acids long for the spike protein, and the RDRP is over 7,000. Yet I was able to find significantly more amino acid sequences that align up with the spike protein than I did with the RDRP, which is 7 times longer. Now, that could be from biases in the way that we do research, because we could do a lot of research on the, oh, how do I phrase that? We do a lot of research on physiologically relevant amino acid sequences. So that could mean that the stuff that we're looking for is just going to so happen be more akin to the stuff that we find on the spike protein, um, or how to phrase that? And the spike protein is evolving to do more physiologically relevant, or have more physiologically relevant epitopes or sequences on them, uh, just because those are active surface types of sequences. And when we do our searches, we're often searching for surface-like amino acid sequences that have some physiological response. And so there could be a lot of overlap just from that type of bias in our research alone. But that's the entire point. The entire point of doing these types of searches is that we find parts, or we find the physiologically or surface related peptides, because that's how antibodies work. Antibodies act upon the surfaces of these proteins and peptides. So what I'm going to do is, I have 1,000 tabs up right now, and I think what might be better is I'm just going to make a completely different video where I go through, I'll go through a lot of these sequences and I'll give my thoughts on a lot of these hits, because some of these hits that I have found could explain some of the differences or some of the pathology of the SARS coronavirus. So granted, it's not all encompassing because we haven't done research on every single amino acid sequence, but out of the sequences that we have studied, there are a few that I could imagine, if an antibody was being produced against one of these amino acid sequences in the spike protein, those would easily be able to cross-react with some very important peptides in the human body, or in the host body. So with all that, I'm going to end this video, and then I'm going to make a new video going through these sequences. So thanks for showing. Leave questions in the comments. If you And remember, this is all hand wavy. This is all just, very, very autistically going into the databases, trying to find any patterns. But I do believe I'm going to justify the uh, the philosophy of this because this is exactly what antibodies do when they are doing their own attacks on protein sequences. And since we know that viruses have evolved a strategy to attack corticotropin, it makes sense to look for um, the amino acid sequences that might align with corticotropin, and then possibly a lot of other hormones or that are in the human body as well. And so I think there was a paper that I read where they went over hepcidin. Yeah, I'm rambling right now, but they went over hepcidin, and hepcidin is one of the very, very small peptides that's associated with I think it's iron metabolism. Yeah, don't quote me on that, but I believe it's iron metabolism that it plays an extremely large role in. And so if you're able to dysregulate I think that was it. Hepcidin is like the insulin of iron. So insulin, if that's being released into the body, that tells your bodies to take sugar out of the blood and put it into the cells. Hepcidin does the same thing, but instead of with sugar, it does it with iron. So um, by producing hepcidin analogs, you could trick your body into either thinking that there's hepcidin, or you could create the antibodies to do the opposite, so that the iron stays in the blood serum. Regardless of what strategy the viruses are employing, that could be something that we need to worry about. Is that and I believe I also did a blast search on hepcidin on the spike protein, and I did find near the end of the spike protein some parts that did look like hepcidin. But the problem is it was a very cysteine rich residue, and for those of you who don't know, cysteine can form covalent bonds with other cysteines, and so a very common motif in biochemistry are these cysteine knots and just cysteine sequences. And so it's very easy if you have a cysteine rich area that a lot of biomolecules are copying these cysteine sequences off of each other, and this very likely has come just by chance. But it is something worth noting. Um, and I should also mention a lot of toxins are cysteine rich pieces, and so when you do these searches, you'll end up if you do have a cysteine rich area of your protein, it could very easily line up with cysteine rich toxins. But yeah, I'm rambling right now, so just go down into the description, like and subscribe if you want to get more updates and more ramblings from me. So yeah, thanks.